Yes, my name is George Foreman III, and this is my presentation for lactate pathways. Lactate pathways. So, this all starts with the fast glycolysis. As pyruvate is a product of the second part of fast glycolysis, which is anaerobic metabolism, which is when oxygen isn't present. As when oxygen is present, pyruvate goes to the Krebs cycle. Let's see. So when oxygen is present, it enters this cycle. So pyruvate can be converted into lactate in the sarcoplasm by the enzyme lactate dehydrogenase, aka LDH. This process also oxidizes NADH to NAD+, producing hydrogen ions, which is also known as lactate acid fermentation. Like I said earlier, this occurs with no oxygen because the pyruvate can't go into the Krebs cycle. And eventually in the muscle cell, NAD+ eventually runs out because it is converted to NADH. You see, here's a picture of the process, pyruvate and NADH. It can go both ways. And then there's a transporter, MCT1, which is on the muscle cell membrane, and this transports lactate out and into the blood. It also is proton linked, so it can also transport hydrogen ions out of the skeletal muscle at the same time. The blood can buffer hydrogen ions from skeletal muscle to prevent the muscle from becoming acidotic. And then when this occurs, the liver also has MCT1 transporters, so the liver accepts the lactate. So lactate in the blood can enter the liver and be acted on by LDH to form pyruvate. This reaction goes in the opposite direction from anaerobic muscle because the liver is not anaerobic. Liver has enough NAD+ levels to form NADH. Pyruvate can undergo gluconeogenesis to form glucose in this step of the stage or in the cycle. And as you can see, it enters the liver, lactate, LDH, pyruvate, then gluconeogenesis to form glucose. See, 2 pyruvates are required to form 1 glucose. That's because glucose is 6 carbon. And pyruvate is made out of 3 carbons. The cost of this step or this stage is 6 ADP, and let's see. And this whole cycle is the Cori cycle. As you can see, here's a simple breakdown right here with just the muscle, the blood lactate, blood glucose. And here's more into depth breakdown, showing which enzymes the enzymes working. The LDH, GAPDH is another enzyme that's more on the glycolysis side, and the blood. In regards to lactate versus lactic acid, this illustrates it's pretty much where is the hydrogen ion, as lactate is a product of lactate dehydrogenase reaction, as I said earlier. Lactate is lactic acid missing 1 proton. I said the proton can go away. And yeah, the proton consumption, proton. Yeah, sorry, of the ion. Not a chemist. And as you see, this breaks it down. So wherever the hydrogen ion dictates if it's lactate or lactic acid. And pretty much like it says, when lactic acid donates its proton, it becomes a conjugate base, lactate. So it's almost like a square rectangle. There you go, switch slides. Let's see, as far as training goes, let's see. Lactate production increases with exercise intensity. That's one of the main factors. Muscle fiber type influences production. type 2 fibers have been seen to have a higher production rate. Some other factors that influence lactate production: exercise duration, state of training, and initial glycogen levels. They all influence lactate accumulation. And some studies have been shown to that both aerobically trained and anaerobically trained individuals have faster lactate clearance rates. Blood lactate is greater after high intensity intermittent exercises. So like I said earlier, the intensity levels of the exercise and the workout dictate lactate levels, and ways of training that can influence resistance training can result in alterations in lactate response. As far as the lactate threshold goes, let's see, it begins at about 50 to 60% maximum oxygen uptake in untrained individuals, and if people are trained, 70 to 80%, especially in aerobically trained individuals. So there's lactate threshold, LT, and then OBLA, which is onset of blood lactate accumulation, which is when intermediate and large motor units are recruited during exercise, during increasing exercise intensities. These usually involve, like I said earlier, type 2 muscles. And as far as training that can influence both these, shifts can occur in the lactate threshold and the OBLA because of changes in hormone release, mainly catecholamines. Let's see, the catecholamines are lowered. There's a lowered catecholamine release at the high exercise intensities, and there's also another training adaptation that influences this is the increase of mitochondrial density, which allows for greater ATP production through anaerobic mechanisms. And that's my one main reference and where I got the pictures from. And like I said, that's pretty much the Cori cycle. Thank you. That is it. Yeah.